Okay, welcome to Marine Invertebrate Biodiversity, the lecture on sponges, phylum Porifera. So we're talking about sponges today, phylum Porifera. I want to give a little bit on basic characteristics of sponges, and then talk about the types of sponges that we see, a little bit on sponge physiology, morphology and physiology, and then a little bit on sponge evolution and where they fit into the other metazoan phyla. So that's where we're going, and we're off. So, phylum Porifera. Phylum Porifera are the sponges. And so porifera comes from Latin. Porus is pore, and fera, F-E-R-R-E, is to bear. So what we have with porifera are pore bearers. So porifera are pore bearers. So these are organisms, metazoans, that bear pores. And that'll make a lot more sense when we talk about their structure. So if we look at number of species, we're looking at around 7,000 to 15,000 extant species. So of those roughly 7 to 15,000 extant species, about 98% are marine. And so we've got a great organism to start with for this course because we've got a phylum that is dominated by marine organisms. So my third point here as a way of introduction is their general habitat, in that they prefer hard substrates. So when you go to look for sponges, hard substrates are where you'll find them: rocks, pilings, reefs, things like that. They inhabit a range of depths from very shallow habitats to the deep oceans, and they're most diverse in shallow waters, for example on tropical reefs. So found over a range of habitats, but most diverse in shallow waters. In terms of size, they cover a range of sizes, but they can be quite large. So often they're rather small, but they can be quite large. And so 2 meters on Caribbean reefs is not uncommon for some really large sponges, as shown here in this image of a diver on the right-hand side. They can also be rather large and rather old under the Antarctic ice. And so about 2 meter tall, 10,000 year old rosellid sponges from Antarctica have been reported as well. So very old, again, around that same 2 meter size. So in the Antarctic, it's an interesting case. There's a lot of silica, which we're going to learn about in terms of how sponges are using for their structural elements, their spicules. Calcium carbonate precipitation is rather difficult in those habitats, but there's a lot of silica, and so they're using that silica for their structural elements, and they can grow to large sizes and maintain that for a long time. So a fifth point here, these are the simplest multicellular animals, the simplest metazoans. And remember, I mentioned them earlier in the last 2 lectures ago, in that the sponge body plan has maintained a rather simple form for a very long time, and it's been very successful. So a nice thing about the sponge body plan is there's no fixed body shape. So there's no fixed body shape. They're asymmetrical. And there's no anterior-posterior axis. So there's no anterior-posterior or aboral oral surface. So there's no aboral or oral surface. Try to erase this, and there we go. So we've got no fixed body shape. There's asymmetry. There's no symmetry to the organism, so they have an asymmetric body form. And there is no anterior, posterior, sort of front, back, or oral, aboral sort of feeding surface, non-feeding surface for these organisms. And so they grow in a variety of ways shaped by environmental influences and where there's space and competition allows them to grow. So if we look at another point here, they have a long evolutionary history. So there are some interesting sponge fossils, and those are shown in these images around the screen. They've been in the fossil record since the early Cambrian. So fossils from the early Cambrian. So we're looking at around 580 million years ago. So quite a long time. And they were very common in paleo seafloor habitats, and so they were quite abundant during that time, and they're still abundant today. If we look at some of the distinguishing characteristics of sponges, and we list them, one is that they're sessile and immobile. And so when you look at sponges, what you see is a body plan that's really well built for this lifestyle. And so they're sessile and immobile, they're not moving around very much, and their body plan works really well in a habitat or in a lifestyle that's very, very static. And so one of the interesting sort of historical notes about that is they were originally described as plants, not as animals, because of this sessile lifestyle. It wasn't until about 1765 that they were recognized as animals and not plants. And they lack locomotory structures. So there are no locomotory structures. They also lack sort of the sensory structure and the motor structure that underlie what we typically find as a locomotory structure. So by that I mean nerves and muscles. So they lack the physical morphological structures for motion, and they lack the underpinnings of that in terms of nerves and muscles. Their body plan is organized around a system of pores. So they're organized around a system of pores. And in addition to that, there are pores, there are passageways, and chambers through which water is going to continuously flow. Through which water continuously flows. And so we've got an organism whose body plan is organized around a system of pores, passageways, and chambers through which water continuously flows. And they're going to be dependent on that water flow for a variety of life processes. So they're dependent on water flow for a whole variety of things, including food, gas exchange, in terms of obtaining oxygen and releasing carbon dioxide from cellular respiration, the release of gametes, so release of gametes for reproduction, and the removal of digestive and excretory waste. So obtaining food, respiratory gas exchange, reproduction, removal of wastes, are really going to depend on the water that they're flowing through their bodies. And understanding sponge architecture is really key to understanding their biology and how they work. So understanding how the bodies are put together and the cell types and how water flows through individual different types of sponges is really important for understanding how all of these processes play out in the sponge. And so if we look in a very sort of simple schematic sponge, we can draw this out, right? So we're looking at something like that, sort of a barrel-like structure, and we have some pores, some holes all over. And so we can look at these different parts of the sponge. If we just look at generally how water's flowing, we've got water on the outside of the sponge. Water is going to go in through this pore network. It's going to move through the tissue and out through the central pore here in this kind of a fashion. And let's go label some structures. And so we've got water coming in. Where water enters are through these ostia. So multiple ostia, an individual ostium, right there. And those are the incurrent pores. So we've got our incurrent pores. And then you've got an osculum, which is the excurrent pore. So the osculum is the excurrent pore. So we've got water entering from the external environment into ostia, individual ostia, and then going into what's termed the spongiocele or the atrium, this empty central cavity, and flowing up into out of the animal through the osculum. And so this empty central cavity is the spongiocele. The spongiocele or the atrium is an empty central cavity where water is going to collect, be pumped through the animal into the center, through all these many ostia, collect in the spongiocele, and then be passed up through the osculum. And so as we go forth through the lecture, we'll look at different types of plays on this general body plan, but this is the general sponge body plan. So if we look at our third distinguishing characteristic here, we've got a structural element, and that is spicules. So we've got these structures called spicules that are going to function as skeletons. I say skeletal elements to support the body. So skeletal elements to support the body. And the spicule size and shape is really an important taxonomic character. So taxonomic character is these spicules represent that. And so for identifying species, it's very useful to dissolve a little bit of sponge tissue with some acid and examine the spicules. And so hopefully you'll get a chance to do that before this course is over. And spicules can be made from calcite, a calcium salt, or silica, a silicon salt. And so calcite or silica are what make the spicules. It's what the spicules are made of. So calcite, a calcium salt, or silica, a silicon salt. And so these are sort of hard structures. The material can vary even among closely related species, and so it's the general structure that the overall structure, three-dimensional structure that tends to be observed in these guys. Here are some examples of spicules. Very strikingly beautiful structures, really fascinating shapes, and so some interesting structures, and just an amazing diversity of shapes when you look at these. So on the left is just a light micrograph, and then on the right is a series of scanning electron micrographs put in composite. So really interesting club-shaped, pointy stars and all sorts of different structures. Really beautiful. So this is figure 4 from the Pechenik text, and if you look at the position of the spicules, it's cartooned in this figure. And so spicules are shown right here. They're all throughout the mesohyal region. So the spicules are in the mesohyal. mesohyal region of the sponge. They're essentially embedded in a cell matrix that's sort of this gelatinous and proteinaceous material with cells. So they're embedded and scattered in a matrix of cells and protein. So proteinaceous material. And that protein is dominated by collagen and spongin, two proteins. So we've got collagen and spongin are the two proteins that dominate the mesohyal. And these are strong, they're sort of squeezable, absorbent proteins that create this outer matrix of the sponge, in which there are spicules to give it some additional structure. They often project through the body wall, and that's cartooned right here. So you can see these spicules pointing out, and what that does is when you look at a sponge, particularly in the water, it has this sort of hairy structure to it, almost like a fuzzy, prickly surface. And that's because the spicules are protruding through the outer body wall. So our fourth characteristic here in distinguishing sponges is their cell types. And so sponges are going to possess specialized cell types. Specialized cell types, not organs. So they're going to lack organs. So instead, they have these specialized cell types that perform a variety of functions. So rather than organs, they have these specialized cell types. Again, there are no specialized organs for reproduction, digestion, excretion, respiration, sensation. They lack those organ structures. No specialized organs in these guys, just specialized cell types that we'll talk about. So when we look at these cell types, the first are pinacocytes. So we've got pinacocytes. These are shown in figure 4. They're also in figure 4.1. They're also in figure 4.5 that I'll show you in a moment in your text. So the pinacocytes are this outer layer. They're forming what's called the pinacoderm. So they cover the outer surface. They cover the outer surface of the sponge, and they form a one cell layer thick outer body layer. They form the pinacoderm. One cell layer thick. Sort of outer body covering of these organisms. There are also basilar pinacocytes. So basilar on the bottom, basilar pinacocytes, and this is where the sponge is going to attach to the hard substrate, and they're going to secrete adhesive material, adhesive proteins, to stick the sponge to the substrate. So these basilar pinacocytes are going to secrete adhesive to attach the sponge to the substrate and maintain the sponge's position in that habitat. So our next specialized. Oh, I should show you this on the image here. So pinacocytes. This is what we're talking about. These pinacocytes right here. So they're laying on this outer body layer in the pinacoderm. Okay, our next cell type are porocytes. So porocytes, you can see in figure 4.1, the porocyte right here, it's this large channel structure. And then there are also porocytes shown right here where their openings make up the ostia. So these are modified, almost donut-shaped pinacocytes. These are modified pinacocytes, almost having a donut-like shape with a central pore. So the central pore is the ostium. So the ostium is that central pore, the incurrent pore. It's the incurrent pore where water enters. Where water enters. And so this is the central pore there, the ostium. The diameter can change to regulate water flow. It's unclear at this point what the mechanism is, but the diameter can change. The diameter can change. It's in the least complex sponges, the porocytes can extend from the outer body surface all the way through the mesohyal into the spongiocele. And that's what you're seeing here in this figure. I believe this is figure 4.5. Yeah, that's figure 4.5. And so, and what you're seeing in this case is this entire distance is maintained by the porocytes. So in these very simple sponges, you're seeing the porocyte actually extend through the entire body wall from the pinacocyte region all the way into the spongiocele. That won't be the case in the more complex sponges. So our third type of specialized cell are the choanocytes. So the choanocytes, these are flagellated cells that are lining the spongiocele. So flagellated cells that line the spongiocele, and they're going to form the choanoderm, again, a one cell layer thick region. Again, one cell thick. And that's what you're seeing right here. So these are the flagella of the choanocytes. Then they have a collar made of microvilli, and then the nucleus in the central portion of the cell. And so they're forming this one cell layer thick region lining the spongiocele. And so the spongiocele, that inner portion of the sponge. If we just were to draw one, it would look something like this. And we've got some organelles inside. So we've got a nucleus I just drew here, and a lysosome with food particles. So a lysosome with food particles, we'll draw another one in. So this single flagellum is going to extend into the spongiocele. And there's going to be an uncoordinated beating of flagella that's going to move water through the sponge. So there's an uncoordinated beating of these flagellae from all the choanocytes that are going to draw water in through the porocytes, through those ostia, into the spongiocele and then out the sponge. If we look at the functions of these cells, these choanocytes, they have three major functions. And so their first major function is to generate currents for water circulation through the sponge. So they're going to generate currents for water circulation through the sponge. Most sponges filter their own weight in water every 5 seconds. So a tremendous amount of water is moving through the sponge. A 10 centimeter Leuconia sponge is going to pump about 23 liters of water per day through 2.25 million flagellated chambers. And that'll make more sense when we talk about the structure of one of those sponges. But it's a large amount of water, a large volume of water in a given period of time for these guys. And so it's the function of these small choanocytes that are actually doing this and drawing all this water through the cell. So very remarkable. These cells are also going to have a feeding function. They're going to capture food particles. So capture small food particles. And they're also going to be involved in reproduction in terms of capturing incoming sperm for fertilization. And so they're going to play these multiple roles. So we look at the sclerocytes next. So we talk about sclerocytes. These are amoeboid cells. That are going to secrete the spicules. So these are these amoeboid cells, sclerocytes, that are going to secrete spicules. And then E, we've got another cell type here, the spongocytes. The spongocytes, and these spongocytes, again, are amoeboid cells that are going to produce spongin. And remember, spongin is this protein, this sort of stretchy, absorbent protein that's going to be present in the mesohyle, the matrix of cells and spicules. And so those are our two additional cell types, and we've got a final cell type that are archaeocytes. Oops. So a final cell type are archaeocytes. These are undifferentiated cells in the mesohyle. So we've got undifferentiated cells within the mesohyle. And again, these are amoeboid in structure, so kind of irregular in structure. And they're going to move by cytoplasmic streaming of pseudopodia, so sort of a typical amoeboid type cell. And so there's a cartoon of one. Curiously, these cells are totipotent. So they're totipotent cells. They can transform. They can transform into any sponge cell type. So these are cells that can transform into any sponge cell type. They could become sclerocytes, spongocytes, choanocytes, whatever. They can also digest and store food particles. So involved in feeding as well. So digesting and storing food particles. All sponge cells can phagocytize food as well. So this sort of feeding role is very generalized in these organisms. These cells, these archaeocytes, also produce sperm and egg. So they produce gametes. Choanocytes can function in this role as well. We'll mention that in a bit. But archaeocytes can play all of these roles. So as we think about these distinguishing characteristics and review them a bit, we said they were sessile. We said that these sponges are organized with chambers for water flow. They have the skeleton made of spicules that are maintained in the mesohyle. And we went through their different specialized cell types. Again, I emphasize no organs. There's a fifth one that I want to mention, a fifth distinguishing characteristic, and that is what we just spoke about, in that some cells are totipotent. So some cells are totipotent. They're going to grow by adding new cells that are going to differentiate as needed. So you maintain this population of archaeocytes that then transform into additional cell types as needed to allow the sponge to grow. Again, archaeocytes are the primary totipotent cells, so they have this totipotent class of cells. We can ask the question, why are sponges so successful? Because indeed they are. They're common, they're abundant, they're very widespread. These are very successful organisms. And one explanation for this has been given in terms of their morphological simplicity. So one reason for this might be that they're morphologically simple. And this echoes a point that I've made before, in that a high degree of complexity isn't necessarily essential for success for organisms, and these are good examples of that. That said, the sponge body plan does allow for some very sophisticated design features. So they have a high degree of morphological simplicity, but they can have some sophisticated elements. Or I'll just say some sophistication. And so this is a building that was designed based on sort of the internal spicule arrangement within the mesohyal. At least it was inspired. So this is a little bit abstract. For a little bit more of a concrete example, these are hexactinellid glass sponges on the left, and then a higher resolution image of one in the center. And an analysis of this, of the structural spicule arrangement in these hexactinellid glass sponges, these are deep water sponges, it was found to have 7 design elements, sort of the way that struts are organized and the scaffolding structure of skyscraper buildings. And so 7 ways that struts are sort of positioned within skyscrapers were found as structural organizational components of these sponges. And there was a group in Bell Labs who did this analysis and then went back and is trying to develop better building structures or biomimetics from sponge architecture. And so sponges have done this on 1,000 times smaller scale than we do with building architecture. And so it's an interesting, this idea of sort of miniaturizing these structural elements is thought to be perhaps a way that sponges can help inform us about how to build better buildings. So another way or reason why sponges perhaps are so successful is a very high capacity for regeneration. So they have a high capacity for regeneration. And so when you separate sponge cells, they very quickly form new sponges. So there's regeneration of new sponges from isolated sponge cells. And so if you mix the cells from 2 species, so if you mix 2 species cells, they'll sort and reform 2 species. And so this is, it's generally believed then that there must be some sort of immunological self-recognition. So determining cells of a self versus a non-self. And this is the topic of Focus Box 4.1 in your text. And so you might look at that, and perhaps a paper that you might want to present to the class could come from that. This regeneration, it's thought that the totipotent archaeocytes are critical for this. So this process of regeneration and growth and differentiation, it's thought the archaeocytes are critical, particularly for the regeneration process. Other minerals are also required, calcium and magnesium, and cell surface molecules to help the cells associate with themselves once they've been separated. But the role of the archaeocytes is thought to be very important. Historically, this idea of cells from sponges reorganizing has been used to culture bath sponges. And so sponges, when natural sponges are used for bathing, sponge farmers would isolate individual sponges and then grow them in certain shapes and be able to multiply their sponge crop by cutting them up and gluing them to rocks and letting them grow. So this is just an interesting historical note. There's an interesting study that we might talk more about in our discussion of this species, Microciona prolifera, the Atlantic red beard sponge. And this is just showing you an image of the animal right here. So you can see this sort of red finger-like set of structures. Down here in the bottom corner, panels A, B, and C are some images from a study by Sabella et al. 2007. And what these authors found was that you could dislodge cells from the sponge and they would immediately reorganize themselves actively. They would actively move to reorganize themselves. So there was sort of an active migration to reorganize when cells were removed. So those cells actively moved to organize themselves. New sponges would form in 2 to 3 weeks. So regeneration time of about 2 to 3 weeks for a new sponge. And interestingly, the original cells seem to perform original functions. So original function of the original sponge. So I'll say original function of original cells in that sponge. And so what these authors are doing, again, they have a bead, which is shown right here, and then they're attaching parts of a sponge to it, and then the sponge is growing on top of that. And then in panel A, they're actually doing a graft of different species. And so what they're doing is showing that the only fingers of a sponge that can be grafted back onto the same species were ones that were from the self. So this again is an idea of immunological self-recognition, again, because only the self can be grafted and attached back on. So an interesting study aimed at this, and there are plenty others. So a third reason why these sponges are so successful is their genes. They actually have a very extensive array of metazoan gene families. There's an extensive array of metazoan gene families. And so they have a very full toolkit of genes to work from to produce a variety of biological functions. They have ancestral biocalcification genes, presumably involved in spicule production and perhaps other things. Carbonic anhydrase genes, alpha carbonic anhydrase, are some of the genes involved in that. They have the production of many secondary metabolites. Secondary metabolites. So again, these, you might be familiar with these in plants or perhaps some other animals. They are chemicals that they're producing to deter predators and allow them to compete for space. And so chemicals to deter predators and compete for space. Again, very common in plants. Again, sessile organisms, so it's not terribly surprising that they do this. An interesting example of this is Siona species. So an example here, Siona species. This is a boring sponge. We have a yellow boring sponge here in Rehoboth Bay and other estuarine areas. And this sponge has amoebocytes. So this sponge has amoebocytes that produce calcium carbonate dissolving compounds. And they use this in an interesting way. So calcium carbonate dissolving compounds from amoebocytes are going to allow this animal to remove fragments of corals, clams, and scallops. And so they sort of bore into these animals, and they use them as substrate too. So you'll find them sort of attached to a very large clam, for example. Mercenaria, they like to attach to Mercenaria, chowder clams, and so you'll just see this big sponge attached as a hard substrate to this large clam, and they'll kind of dissolve that sponge over time to hold on. Some of these secondary metabolites are also of human benefit as well. So Maoris in New Zealand used potassium fluorosilicate, which was derived from sponges, as an anti-inflammatory drug. And there's quite a bit of interest, although not a huge amount of success, but quite a bit of interest in anti-cancer drugs from deep sea sponges. And so there's quite a bit of that interest. Again, not a huge amount of success, but a lot of potential. And a fourth way that sponges are so successful, or reason for their success, is their mutualistic association with microbes and algae. Let's see, let's erase that. So there's a mutualistic association with bacteria and algae. And in this case, you get the sponge serving as the host, which is going to provide the bacterial or algal symbiont with a place to live. And the symbionts are going to translocate primary metabolites or secondary. So primary is going to be food, or secondary metabolites involved in defense to the sponge. So the sponge is generating sort of a primary food source from the symbionts, and as well as secondary metabolites involved in defense. So a lot of the defensive capabilities of sponges are actually coming from their symbionts, not compounds that they're directly producing. So what you can do in this case is you can really add to the biochemical repertoire of the host through the metabolic capabilities of the symbionts. And this is a very common theme that's starting to emerge throughout biology. And so the symbiont is often as important, if not more so, than the host in a lot of ways for the host's biology. Symbionts can make up to half of the sponge volume. So up to half of the sponge volume can be symbiont. So a tremendous amount of the sponge is actually the symbiont. Cell surface proteins of microbial cells are often allowing them to resist being phagocytized by sponge cells and allowing them to maintain themselves within a sponge, so their cell surface proteins are being recognized by the sponge cells. And it's thought that perhaps some symbionts are transferred in eggs and larvae, but that's not quite clear, but there's some evidence for that. So symbionts are very important, as they are in cnidarians, and we'll talk about that probably the next time. Okay, when we look at sponge morphology, we see that three main morphotypes have evolved. And so there are three main morphotypes of sponges: the asconoids, the sechanoid, and the leuconoid sponges. And I'm going to go through those now. They're going to differ in a few substantial ways, and primarily a difference is the extent that the choanocytes are evaginated from the spongiocele. So they're going to primarily differ in the extent that choanocytes are evaginated from the spongiocele. So the degree to which that they're separated and sort of within the sponge body structure relative to the spongiocele. What this is going to do is change the flagellated surface area. And so they can dramatically increase the flagellated surface area. They can increase flagellated surface area with evagination. And what that's going to do is it's going to alter current flow. So it's going to alter the current flow through the spongiocele and the amount of surface area that's available for food collection. So by increasing the evagination, by having all these little chambers, which you're seeing here and to some extent here. So this is going to be simple. This is more complex. And this is even more complex. As you go from asconoid to sconoid into leuconoid sponges, you're increasing the degree of evagination and then increasing the flagellated surface area. And so that's what you're seeing with this changing morphotype. And so if we start out with the simplest ones, the asconoid sponges. So asconoid sponges have a simple body structure. So this is our asconoid sponge. A simple body structure in these guys. There's a large central cavity. So there's a large central cavity that's lined with choanocytes. And the pinacoderm is going to lack folding, so it's just a smooth pinacoderm surface. Pinacoderm is going to lack folds. So it's just a smooth pinacoderm surface. And water flow is going to look like this. It's going to have ostia into a flagellated spongiocele, into the osculum. So if you were to follow water flow through one of these sponges, you're going to go inside from the external environment through into a flagellated spongiocele, so through the ostia right here, into a flagellated spongiocele, and then out the osculum. And so that's the general path of water in these guys. The problem with this general body design is really one of what happens when you grow larger. And it's a geometric problem. So growing larger for asconoid sponges means a disproportionate increase in spongiocele volume. So growing larger is bad because the spongiocele volume increases disproportionately to spongiocele surface area. And if you think about that, volume increases as a cube to the third power, and surface area increases as a square to the second power. But flow is proportional to surface area and not volume. And so these sponges tend to be small. The problem by having this really large spongiocele is you don't pass water through the sponge as readily. You're increasing your volume much more quickly than you are the pumping power that you have in your flagellated spongiocele. And so what you see in sort of the sponges that are asconoid sponges is that they tend to be rather small. And so there seems to be a size limitation. So they tend to be small. So our second sponge type are the coenoid sponges. So our coenoid sponges. They're more complex than asconoids. So they're slightly more complex. What you see is some folding of the pinacoderm. So the pinacoderm is pushed out. And it's pushed out into hundreds of finger-like projections. So you have all of these little fingers that are being pushed out, and that's shown right here in this cartoon of our coenoid sponge. And so what you end up with when you have these projections are what are called incurrent canals and radial canals. So you have incurrent and radial. Well, I'll just do it this way. So you have incurrent canals and radial canals. And your incurrent canals are the invaginations of that outer pinacoderm. And the radial canals are the folds, so the invagination of the outer pinacoderm. The radial canals are the folds on the inner pinacoderm. And I'll draw a cartoon of that on the next slide to try to make that a little bit clearer. So you have these incurrent canals and the radial canals. And so to kind of cartoon that, if you were to imagine these folds kind of go up, and then there's going to be some gaps here, right here, and these are going to be the ostia. Right, and so you've got these folds, and so these spaces here are the incurrent canals, and these spaces here are the radial canals. And the choanocytes are going to line the radial canals. Choanocytes line the radial canals. And all of these little gaps are ostia. And so you've got water coming in, being pumped by the choanocytes lining the inside of the radial canals. So the radial canals have these choanocytes. What this design is going to allow is reduced spongiceal volume. So this is going to allow for reduced spongiceal volume because of this added radial canal volume. You have all those choanocytes lining those radial canals, and you don't need them on the spongiceal. And so you can have a smaller spongiceal for a lot more pumping power. And so that's what you see. This is really the same trick that you see in guts of other metazoans, where you want to increase surface area for digestion. You want to increase surface area for pumping in the sponge, and what you do is you increase the surface area by all these folds, creating these radial canals. So this is a pretty common trick. So reduced spongiceal volume because of this added radial canal volume, and also increased efficiency of water flow because the choanocytes are concentrated in the radial canals. So there appears to be some increased water flow efficiency. So we'll just call it increased efficiency of pumping because of this concentration of choanocytes. So when you look at the water flow pattern through a coenoid sponge, you've got incurrent canals and then ostia. This is kind of where a water parcel passes. Incurrent canals, ostia, the flagellated radial canals. Flagellated radial canals into a reduced spongiocele. And then through the osculum. So this is the pathway: incurrent canals through the ostia, flagellated radial canals into a reduced spongiocele, and then finally out the sponge through the osculum, and that's shown right here. So we're going in through the incurrents, ostia, radial canals, spongiocele, which is reduced, and then out the osculum. So our last sponge type is the most complex, and these are leuconoid sponges. These are leuconoid sponges. They're going to have the most pinacoderm folding of the morphotypes. The most pinacoderm folding of the three morphotypes is found in these guys. And so the incurrent canals are very internal and they're very branched. So the incurrent canals are internal and branched, so even more surface area getting into the radial canals. So you've got this internal network of incurrent canals, and then you've got a radial canal structure. And so the radial canals have changed a little bit too. So the radial canals are small, round, flagellated chambers. So they're small, round, flagellated chambers, and the spongiocele is even more reduced. Again, the spongiocele was large in our asconoid sponges, largest in our asconoid sponges, because that was where the choanocytes were, and that's where the surface area was required for pumping. That became reduced in the ceconoid sponges as we had these radial canals and concentration of flagellated cells, and now that we've got our leuconoid sponges, they're even more kind of concentrated and replicated as these small flagellated chambers. So this continual reduction of the spongiocele as it's become less critical for generating the surface area that generates flow. So water is going to enter ostia and then get into this network of passageways. And then it's going to pass into branching incurrent canals, as I mentioned. And then, so that's what you're seeing right here. So these are ostia, water flow into this branching network of incurrent canals. And then those canals are going to open into the flagellated chambers. Through what are termed prosopiles. Through prosopiles. And these prosopiles, again, are just the opening of the flagellated chambers. So it's where the arrowhead is right here, these prosopiles. And then water is going to enter excurrent canals. Excurrent canals, which increase in size as these canals join. And so you're seeing this pathway of water in through in through ostia, in through the incurrents, through these radial canals, and then into these excurrent canals. And then those are going to increase in size as water ends up leaving the sponge through oscula. And so when you think about the sponges, a typical sponge that you see is actually going to be this leuconoid type. So about 90% of the extant sponge species are leuconoid sponges. About 90% of the extant species. And these are going to be in class Demospongiae. So if you see a sponge, the odds are it's class Demospongiae and it's going to be a leuconoid sponge. This structure isn't always obvious. This incurrent canal and all these incurrent, excurrent canals and the prosopile type structure isn't always obvious, but they're there nonetheless. This suggests just the abundance of this morphotype suggests it's most efficient. It can grow to larger sizes because of the number of flagellated chambers and how these can just be increased as the body size grows. So you're not dependent on a very, very large spongicele, but rather you can have a lot of pumping through all of these chambers. Typically, several oscula are present rather than a single osculum. So often several oscula rather than just a single osculum. So if we look at the general water flow pattern as we've laid out for the others, if we look at water flow, we've got ostia, incurrent canals, prosopiles, again, which are just these pores into the flagellated chambers. So similar to our radial canal structures, and then into the excurrent canals, and then an osculum. And this is shown right here again. We'll take a look at an interesting video of that when we meet up to discuss this material. So those are the sponge morphotypes. We're going to talk a little bit about sponge function at this point. So sponge function, as you've probably guessed, a major aspect of sponge biology is regulation of water flow. And it's dominating our sponge morphotypes. And so we want to talk a little bit about that here. So regulation of water flow. Flagellar activity from the choanocytes is really driving that water flow. So the activity, the beating of the flagella, activity or beating of the flagella is going to drive water flow through the sponge. Feeding is also dependent on water flow through these flagellated chambers. Remember, the choanoflagellates, choanocytes are generating this water flow. They're also capturing particles of food. And so they're flowing water through the flagellated chambers is important. So feeding depends on water flow through the flagellated chambers, or across flagellated surfaces in the case of the asconoid sponges. Sponges are suspension feeders. So they're going to be feeding on particles that are suspended in the water. They're eating typically microscopic planktonic organisms, primarily bacteria and protists. So suspension feeders on plankton, typically bacterial plankton and protists is what they're generally feeding on. And they're, again, the role of water flowing through the sponge is critical. Sponge morphology causes an interesting effect on water velocity. Just based on the degree and the number of flagellated chambers, water velocity is low in the flagellated chambers, and it's much higher in the osculum. And this, again, is an effect of sponge morphology, and it's similar to what happens in your capillaries. And so Sponge morphology is going to cause water velocity to be low in the flagellated chambers. And high in the osculum. And again, this is the same concept as having slow blood flow through your capillaries and higher blood flow through your arteries and veins. And the idea here is that a given fluid volume is going to move slower when it flows through a larger cross-sectional area. And so the idea is that fluid volume, I'll say a fluid volume, so a parcel of fluid is going to move slower when it flows through a larger cross-sectional area. And so one thing that might help you to think about this is think about a river versus a stream. So a river has a nice large cross-sectional area. The area in cross-section of a river is larger than the area in cross-section of a stream. So when the water spreads out from a stream to a river, it has a slower flow. So very high flow in a stream when you're forcing the water volume through a narrow passageway, but when it can spread out in a river, it slows down. And so this is a function of the total cross-sectional area, not the cross-sectional area of an individual element, but the total cross-sectional area in the organism. And so a numerical example of this is given on the next slide. So this is table 4.1. And what you see here are ostia, flagellated chambers, and the osculum. Individual cross-sectional area. The ostia and the flagellated chambers, both very small. The osculum, somewhat larger. The approximate number per sponge is shown right here. And if you multiply individual cross-sectional area by the approximate number per sponge, you'll get a total cross-sectional area, which is shown right here. And so if you look at total cross-sectional area, the largest number, 203, is in the flagellated chambers, right? Also, look at the water velocity here. It's by far the slowest, 10 to the negative 4 centimeters per second. And so flagellated chambers have the largest total cross-sectional area. Therefore, the slowest flow rate. Again, the same trick that's played by your capillaries, small vessels with large total cross-sectional area, therefore slow flow. The osculum has the smallest cross-sectional area and therefore the highest flow rate. So here's water velocity there of 5.1 centimeters per second. So an example from a typical marine leuconoid, looking at how regulation of water flow is a function of the morphology of the sponge. Environmental water flow can also play a role. So fluid flow in the marine environment also matters, and it can be used by the sponge to supplement flagellar flow. So fluid flow in the marine environment can supplement flagellar mediated flow. And so you can have faster flow in faster current. The sponge opening, it can also open ostia that face the incoming current and close ostia on the other side. It could just be faster flow in a current that's generated, so that could just help physically drive flow through the ostia. You can also see opening ostia facing flow and generating more current on that side. So both of those can be observed. It's unclear the role they play in the ecology of sponges, but they've both been observed. So a little bit more on function. An interesting thing that you see is there does appear to be some communication between cells, even though sponges lack sense organs, nerves, and muscles. So there's some communication among the cells, even though there are no nerves, muscles, or sense organs of any kind. And so why that is sort of how this happens mechanistically is an open question. Oscula can close. So oscula, we'll say, can respond to touch, air, toxins, and they can close in response to that or open when those stimuli are removed. So they can certainly respond. Reactions are slow, often with minutes of delay, but the response is still there. Again, it's unclear what the mechanisms are. There are amoebocytes that surround oscula that have microfilaments and microtubules, these contractile proteins in other animals that may serve a contractile role in the oscula, and there's been a few studies on neurotransmitters that may be active in those cells. But there are no nerves or muscles, and so it's an interesting set of questions that underlie that. Let's talk a little bit about reproduction in sponges. And so sponges can reproduce asexually by fragmentation. And this is just when cells break off, as we've mentioned before, or by the production of gemmules. Again, another asexual method. So fragmentation, where cells are just breaking off, or by gemmules, where packets of several cell types are packed up. They're primarily archaeocytes, but other cell types too, and they're packed in a protective coating. And this is what you see down here. This is a gemmule, where you've got sort of this spicule coating with a mass of archaeocytes inside. Again, an asexual method where this is just sort of sent away, and it's resistant to desiccation, anoxia, freezing. So it's a way to sort of protect these archaeocytes until a more favorable habitat presents itself, and then a new sponge will form from them. Mostly freshwater sponges do this, and so it's not really a marine strategy, but we should mention it anyway. So in addition to asexual reproduction, there's also sexual reproduction. We've mentioned gametes, sperms, and eggs, which are broadcast spawned. So there's broadcast spawning. Of the gametes. So these gametes are just released into the water column. They're broadcast spawned. The sponge looks like it's smoking. That's what this picture in the upper right is showing you. Sponges are sequential hermaphrodites. So they're sequential hermaphrodites, and so you'll have an individual that's going to release both sperm and egg, but not at the same time. So not at the same time, but they're capable of releasing both sperm and egg, and they rarely cross-fertilize. So you typically have gametes of different individuals that are fusing. So you've got sequential hermaphroditism without self-fertilization. What you've got in terms of how the gametes are produced, sperm are primarily from choanocytes. So sperm primarily arise from the choanocytes. The eggs are going to be coming from the choanocytes and the archaeocytes. And so you've got both of these cell types producing the gametes. The fertilization process can either be internal or external. So fertilization can either be internal or external. External is in the water column. Internal, the sperm are going to be captured by the choanocytes. So sperm are captured by choanocytes and brought to the mesohyle. So they're brought to eggs in the mesohyle, in which case fertilization is internal because it's happening inside the organism. So when we think about sponges in terms of their relationships, evolutionary relationships to other animals, multicellular animals are thought to have evolved from flagellated protozoa. And so, let me put that this way. So flagellated protozoa are thought to have given rise to metazoa. So our multicellular animals are thought to have evolved from these flagellated protozoa. And the reason for this thought is that all metazoa have a flagella, or have a flagellum at some point in the life cycle. And so I've got down here an image of a sperm. So the male gametes, for example, have a flagella. And so you see this in all metazoa at some point. There's a flagellum at some point in the life cycle. And so this is one piece of evidence for that. And then the sponges and their biology sort of help form another, in that there are choanoflagellates, a type of protist, and there's morphological and genetic evidence that suggests these choanoflagellates, which are shown right here in this micrograph, to be the basal ancestors of metazoans and potentially to have given rise into sponge choanocytes and then so forth through evolution. So there's some morphological and genetic evidence that choanoflagellates, which are protists, are basal ancestors of the metazoa. And choanoflagellates resemble choanocytes. They have one flagellum, they have a collar of microvilli. There's several groups that are interested in this question. One is Nicole King at Berkeley, who's done some really nice work in both the morphological and genetic realm to kind of get at this question. Choanoflagellates are considered to be the sister group of all metazoa, and so they're very interesting in that regard, and they resemble these choanocytes that we've been talking about from sponges. So if we look at an early metazoan cladogram, it would look something like this. So we've got flagellae, choanoflagellates, collagen, this structural protein that we mentioned from sponges, as well as spongin, porifera that we've been discussing, and a nice synapomorphy for them are spicules. There aren't true tissues until after Porifera. Porifera lack true tissues, and organs for that matter, but they lack true tissues, but all other metazoa have true tissues. So all other metazoan taxa, all other metazoa would be at this end of the cladogram. So this would be an early metazoan cladogram showing the position of choanoflagellates, phylum Porifera, a few synapomorphic characters, and then all other metazoa. So that's going to do it for phylum Porifera.